to the Art of Adventure. This is 5 Minute Friday, and I'm going to tell you what it was like to discover a new species. Some of you may know that I used to be a scientist. I was in grad school studying viruses in the hot springs in Yellowstone National Park, and I discovered a new species of virus that's completely unrelated to other viruses or other forms of life. And I recently interviewed my academic mentor and advisor, Mark Young, and he talked about his discoveries. And I wanted to tell you a little bit about what it was like for me. So I'm sitting there in a dark microscope room looking in the electron microscope, and I'm scanning around basically up and down, right and left, looking at this very thin gold-coated surface at the nano micron level. I can't even remember how much I was zoomed in. And I was looking for geometric patterns. That would be the signal of a virus. And all of a sudden, into my field of view, there's just like polka dots full of hexagons. And I jumped out of my chair and I was like, oh my God, this is it. This is something. This is a virus. I've discovered something. And I knew right then that I had discovered something new. That was a single brilliant, exciting moment that was only possible with a year and a half of preparation and was only provable with another year and a half of research to prove it. So I'll talk a little bit about the lead up and the aftermath of that single moment. So when you see something and you know it's a new discovery, well, you still have to prove it and you still have to characterize it and describe it and learn more about it. So first of all, discovering it was this big process. We designed an experiment explicitly to discover new species. And so we went out to over 100 different hot springs and we took samples from all these different hot springs. And then I spent over a year trying to grow microorganisms from the hot springs. And this is actually really hard because we're really good at growing microbes that sort of live at body temperature. You know, you can just stick them in an incubator on a petri dish and they grow. But when you're trying to grow something in boiling acid that, you know, breathes sulfur and iron instead of oxygen, it's like gardening orchids or something. They're really hard to grow. So it took me a year to learn to grow these little organisms. And then, I don't know if we're going to see any viruses inside them or not. But because viruses are associated with pretty much all life, we were crossing our fingers. And what I was hoping was that I would catch some virus particles moving in between cells. So when a virus infects a cell, it makes more viruses. And then somehow it has to leave that cell and go infect some other cells. So I was hoping to catch them sort of floating around. Anyway, once I was able to grow them, I had to concentrate the living material from the vials that I was growing it in. And so I would have maybe just enough to actually be able to see under the microscope. So it took me a year and a half of all these different steps to have a sample that I would even begin to think I can maybe find something here. And so then it was just a process of going and spending 6, 8, 10 hours a day in this microscope room screening through these samples. And most of the time I was just looking at little specks of dirt or bits of broken cellular material, and I wasn't finding anything. But because I had seen viruses under the microscope before, and because I knew sort of what I was looking for, again, viruses they're like an M&M. Their shell is made of proteins, and then inside there's genetic material. And so I was looking for the soccer ball type shape or football type shape, or even like a rod or tube type shape that would signal to me, okay, this is a geometric form that's probably created by a virus. And the virus that I ended up discovering is an icosahedron. So kind of like a soccer ball, but then it has a little ring around the outside. So there's kind of two different interesting things geometrically. So you can really tell when you're looking under the microscope for this thing. And so after I saw it in my microscope, I quickly took a bunch of photos, and there was just lots and lots of these virus particles, which is great. But then I had to take them to my colleagues and my advisors, and we all looked at them together like, oh, does this look like anything existing already? Because if you discover something that's already been discovered, then you don't need to need to bother investigating it some more. And it was looked different enough that we decided to characterize it, decided to describe it. And one of the most important things is to see what the genome is, because once you understand the DNA or the RNA of the genome, you can compare it to all other known viruses, all other known forms of life using computer programs. So that was part of what we did. We were looking at the genome to try to understand what this organism was. And at the same time, we were trying to describe and characterize what the actual particle, the container, the icosahedral structure that I saw under the microscope. So we're doing that by looking at it in different ways, using different scientific techniques to break apart the structure and see if we can find out how big the individual pieces are that come together to make it. Also using 3D imaging technology at a very small scale to sort of understand what the three-dimensional object itself looks like. And so all this, it takes a very long time to actually get usable information to sort of characterize, like if you were to see an animal in the wild, if you're the first person to see, I don't know, a tiger, then you could say, oh, it has stripes, it has these teeth. It's like, here's the environment it lives in. You can sort of look at it and just know right away a lot of things about it. But you don't know necessarily how many bones it has, or what its DNA says, or how long it takes for a tiger to grow up. You have to observe and spend time sort of understanding the finer points of this organism, and that's what we did with this virus. And so it's emotional roller coaster when you see the thing right there in front of you. I mean, there's nothing like that feeling of knowing you're the first person to look at this, of just this knowing, this intuition, like, oh my God, I found something new. This is amazing. To the, just the long hours of manipulating things in the lab to try to get some evidence. And, maybe it takes like 3 months to try to learn a technique, and you fail for 3 months until you finally get a new piece of evidence. And that's what a lot of people don't see in research, is these like months of trying things, trying to grow things, trying to see things, trying to analyze things, and having it not work. And like, okay, is it not there, or is my technique wrong, or do we just not have the technology yet, or what's going on? So to find 10 good pieces of information takes so much work. And that was really hard for me. That was hard to be so patient to try to get some understanding. And I think for people who are not scientists, if you read about a new discovery, it's amazing. Like, there's really no way you can conceptualize how much work went into that behind the scenes. And as you guys know, I left science to become a podcaster and an author and a business coach and travel the world and run Adventure Quest trips and all these other things that I do, which are super fulfilling. And I'm really glad that I had the experience of working as an academic scientist, as a researcher, because there is such rigor and such unknown, and it's really a case where you have to put in a lot of work, and it might not even pan out. And I think having that long-term focus is really, it translates well to almost anything else. I also, as many of you know, do some cycling and endurance sports, and it's very similar in the sense that you can train for several years and you still won't hit your peak of your ability. And this long-term thinking, this long-term projects is way different than the sort of immediate gratification social media age. Like, building something over the long haul and asking a question and then developing ways to answer it, and then like really going deep in research and testing things out, and designing good experiments and sharing your work with peers. Like, there's so much great things to take from the world of research, from the world of academia. And, the other best part of it all was just going out into Yellowstone and scooping up the water from those hot springs in the national park. I mean, just going out there, like, of course it's cool. Like, set up an experiment where you have to go adventuring. I mean, that's why I was in the game in the first place. And then all that time in the dark microscope room, I was like, oh, this is what I didn't see before I started in this process of grad school. All right. Hopefully that was interesting for you. You may not get the chance to discover new species, but I hope you do get to discover something for yourself. So remember, never stop adventuring. Get out there, go explore, design experiments for yourself, ask the big questions. Thanks for listening, guys.